Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. Okay, if you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, we shall start today's class. In the previous video I have discussed about the Microsporangium, stamen. We have discussed about this. Today we shall continue the same with the pistil, Megasporangium, ovule and an embryosac. Okay, as you know, the pistil is the female reproductive part. Okay, the pistil is present in a gynoecium. As you know, there will be a gynoecium. Gynoecium is a female reproductive part, whereas androecium is male reproductive part. Okay, this is male and this one is female. Okay, in a plant gynoecium it consists of a pistil, something like this. Now this pistil consists of three parts, that is stigma, style and ovary. This ovary, it consists of ovule. Okay, stigma, style, ovary, and inside the ovary there is presence of ovule. Now as you can find here, the gynoecium represents the female reproductive part of the flower. Is it? In a flower, gynoecium is a female reproductive part. The gynoecium may consist of single pistil. Gynoecium can consist of single pistil or it can have many pistils. If it is having single pistil, we call it as monocarpellary. Okay, monocarpellary, single carpel. Pistil is also called as carpel. So if gynoecium is having only one pistil, we call it as monocarpellary. If it is having many pistils, then it is called as multicarpellary. Okay, if it is having many pistils, as you can find, or may have more than one pistil, multicarpellary. Okay, when there are more than one pistils may be fused. When it is multicarpellary means, as we said, more than one carpel, and when there are more than one carpel, those carpels can be fused or those can be free. Okay, those carpels can be fused and they can be free. If they are fused, then we call it as syncarpus. Syn means fuse, syncarpus, and if their carpels are free, we call it as apocarpus. Okay, multicarpellary, which consists of many carpels, and those carpels can be fused with each other or they can be free. If they are fused, we call it as syncarpus. If they are free, we call it as apocarpus. Okay, that information you have here. Each pistil, it has three parts: stigma, style and ovary. Stigma serves as a landing platform for pollen grains. Okay, as we have shown, this is a stigma. If you enlarge that part, it will be something like this. This will be a stigma. Stigma is used for the landing portion for pollen grains. Pollen grains will land here, so it is used for the landing platform for pollen grains. And then comes the style. It is elongated, slender part beneath the stigma. Is it? It is long, elongated, and it is slender. Then you can find the basal part, which is the basal part of the pistil is nothing but the ovary. Okay, inside the ovary there will be ovarian cavity, and that cavity consists of ovule. Okay, and in this ovarian cavity there will be presence of placenta. Placenta is what? It is nothing but a tissue. Okay, it consists of which tissue? Parenchymatous tissue, which means it consists of parenchyma cells. Okay, parenchymatous tissue is present in the placenta. Now in this placenta, if we draw one of the ovary, TS of ovary, this one is axial placentation. If you have studied about the first year placentation, or if you remember that, you can find axial placentation. Axial placentation is something like this, right? In this axial placentation, this is placenta. Okay, the placenta is nothing but the tissue which is present here, the cells which are present here. In this placenta there is presence of ovules. Okay, ovules are present. These are ovule, and this complete one, what we call it as ovary. It is called as ovary. Ovary consists of placenta. In placenta there is presence of ovule, and here you can find some seeds, some plants, some ovary will have one ovule or it can have many ovules. Okay, one ovules or many ovules. Now this is having many ovules or one ovule? It is having many ovules. Okay, like a basal placentation, in that you have seen only one ovule was present in something like this kind of structure. If you find in your first year textbook basal placentation, only one ovule is present, whereas here you can find many ovules. Okay, as we know, ovary will, after fertilization, ovary will develop into a fruit and ovule will develop into a seed. Right, after fertilization, ovary, this is the ovary outer covering, this outer covering will become a fruit and these will become it as seeds. If you remember this one in your orange, you might have 8 orange fruit, and in that orange fruit it appears something like this. When you take a transfer section, or when you cut that one, you will find this kind of structure, right? So this one will be the ovary before fertilization, which has turned into a fruit after fertilization, and the seeds which you find at the center. Okay, you can imagine about lime, lemon, or this orange juice, orange fruit. In that you will find these will be the seeds. The seeds are nothing but ovule which were present before fertilization. After fertilization, these ovule have been converted into seed. Okay, so if they ask whether this fruit or this will have many ovules or one ovule, you can imagine about a fruit and you can say whether it is having single ovule or a many ovule. Okay, as in the next page you have rising from the placenta are the megasporangium. Megasporangium is another term for ovule, what we said just now. As we said microsporangia for stamen, here you can call it as megasporangia for ovule. The number of ovules in an ovary may be one, is it? It can have only one ovule, that is wheat, paddy, mango, or it can have many ovules like papaya, watermelon and orchids. Papaya fruit, you might have seen, it consists of many of the seeds. Watermelon consists of many of the seeds. So all these seeds, all these seeds are nothing but the ovule which were present before fertilization. After fertilization, those ovules itself have been converted into seeds. Okay, these are some examples. These can be helpful for your entrance exam. Okay, now they have given some pictures here. So this is a pistil of a hibiscus flower. So you can find there is a stigma, a landing portion for pollen grain. Then there is a slender long style, and then comes the ovule. Inside the ovary, it consists of ovule, and there is a enlarged part called as thalamus, and then there will be a pedicel. Okay. Then they have shown second one, it is multicarpellary syncarpous pistil. Multicarpellary, which consists of many carpels. Syncarpous means many carpels are fused. Okay, pistil of papaver. Next, a multicarpellary. This is also many multicarpellary, or many carpels, or many pistils. So each one is the pistil here. Okay, so you can find many pistils are there, and it is apocarpous. They are free, they are not attached with one another, they are free. So this one is the gynoecium of Michaelia plant. Next comes this diagram. This diagrammatic representation is very important. It is a diagrammatic representation of VS of an anatropous ovule. Here you will find it is an anatropous ovule. This anatropous ovule diagram is very important. They will ask this one for 5 marks to draw and explain, or only to draw 2 or 3 marks. Right? So how to draw this we shall see. Okay, first it will be something like this. Okay, fine. This is how you are going to draw. Okay, roughly I am showing here. So in this you will find there is presence of funicle. This portion we call it as funicle. Okay, then comes this portion called as hilum. Then comes this open area called as micropyle. Okay, now funicle is what you can find here. The explanation of this structure, as many of the students were asking about the notes, how to read, you can give the notes as I used to give earlier in first chapter. but as here you find these are very lengthy chapters and it have lot of explanation for 5 marks, which I could not write and again make a video. It takes lot of time, so that's why what information has been given in your textbook, that itself will work. And even we have in your previous classes or your senior students also have studied about the same, so the textbook itself will act as your notes, just if you refer that one properly. So the Megasporangium or an ovule, okay, about this structure which we have mentioned here. So in this you can find the ovule is a small structure attached to the placenta by means of a stalk called as funicle, okay, which means funicle is used to get attached to the placenta. So here we have shown these are the ovules, is it? So if I draw it once again, if I am showing this is the ovule, it will be something like this, okay. So when it is like this, then it is getting attached to the placenta through what? Through funicle. So this is the funicle which is helping that ovule to get attached to the placenta. Then about funicle you have next, the body of the ovule fuses with the funicle in the region called as hilum. This is the body of the ovule is getting fused with the funicle where? With the hilum, okay. Thus one or two this hilum represents the junction between ovule and funicle, is it? So it is the junction between ovule and a funicle. Each ovule has one or two protective envelopes called as integuments. Integuments are this. If we show it here, these are the integuments, okay. Integuments, how many integuments? 2 integuments are there, 1, 2 and 1 and 2. These are the 2 integuments and here also 2 integuments. What is their function? Their function is protection, okay. Their function is protection. For whom it is protecting? It is protecting for the embryosac. Inside you will find embryosac. It is protected by these integuments. This one is outer integument and this one is inner integument. Okay, outer integument and inner integument. Then you will find this center one. It is called as embryosac. Okay, now this embryosac, it is covered by nucellus, the space which you can find between outer and inner after outer and inner integument, this space which I am just shading here, this part we call it as, okay, nucellus. It is called as nucellus. So this nucellus is what? It is mass of cells which with the food reserve material, okay, which means it is providing nourishment. Its function is to provide nourishment. For whom it will provide nourishment? For the growing embryo sac. As we said, for the pollen grain, tapetum will give this one nourishment, is it? Tapetum, the innermost layer of microsporangia, that will give the nourishment for the growing pollen grains. Here nucellus will give nourishment for the growing embryo sac, okay, or for the growing megaspore. Okay, nucellus, it is nothing but mass of cells with the food reserve material. Function is nourishment, okay. Then here we have said the open part, as we have shown, integuments are not completely covered here on one end, the other side it has been completely covered. The one which is open part of that integuments is called as micropyle. Opposite to that micropyle there is a region called as chalaza. Opposite to micropyle, what end we have? Chalaza or chalazal end or chalazal pole, okay, you can call by any of the name. So it has these parts. First one is funicle, okay, which is used to get attached to the placenta. Then you have a hilum. Hilum is the junction between the body of the ovule and the funicle. And then you can find there is presence of embryo sac at the center. This embryo sac is covered by 2 layers of integuments, and outer integument and inner integument. Their function is protection. Then you can find there is a mass of cells surrounding to the embryo sac. These mass of cells, they are helping for the providing nourishment for the growing embryo sac. And the open part of the ovule we call it as micropyle. Opposite to that, what end we have? It is called as chalazal, okay. So now you can find that information is completely present here, okay. For explanation of that, you can start from here. The ovule is a small structure attached to the placenta by means of a stalk called as funicle. About funicle you have, about hilum you have, integuments, micropyle, chalaza. For all those, all information you have. About nucellus you have, and one more. Located in the nucellus is the embryo sac or a female gametophyte. This we said it as the center one, we said it as embryo sac. It is also called as female gametophyte, Okay, so this female gametophyte, it will an ovule generally has a single embryo sac formed from megaspore to reduction division. Anyhow, from here till here, if you give the explanation of the structure of an ovule, it will be very easy for you for explaining. Okay, so this one is ovule. It is also called as Megasporangium. It is Anatropous ovule. Here different types of ovules will be there with a different shape. Here you can find it is of this shape. This shape we call it as Anatropous ovule. Different shaped ovules will be there like this in this shape. I am just roughly showing like this shape. It might be sometimes the ovule might be of this shape with the integuments with here. So this different kinds of ovules are there, but for your syllabus it has been given only about Anatropous ovule, because most of the enzymes will consist of Anatropous ovule. That's why this has been mentioned. Very important diagram. It has been asked for 5 marks with explanation to draw, only to draw 3 marks or 2 marks. Okay. Next, here you can find next is about Megasporogenesis. As we have studied about first the structure of Microsporangium, then we have studied about Microsporogenesis. Here we shall first we have discussed about structure of Megasporangium, then we are discussing about Megasporogenesis. As we said, first there was presence of the sporogenous tissue, later it has formed pollen grain. Now this embryo sac, how does it form the egg cell inside it? Okay, Megasporogenesis, it is nothing but the process of formation of megaspore from megaspore mother cell. As we said for microsporogenesis, the process of formation of microspore from microspore mother cell is called as microsporogenesis. Similarly, the process of formation of megaspore from megaspore mother cell is called as megasporogenesis. Okay, ovules generally differentiate into single megaspore mother cell in the micropylar region. This is an ovule. Now this is the embryo sac. It consists of single megaspore mother cell. Now this is single megaspore mother cell. Okay, it is nothing but a meiocyte. Meiocytes are always diploid. Okay, now the cell which is present here, it is a meiocyte. It is also called as megaspore mother cell. It is diploid and it is present towards the micropyle and not towards the chelazal end. You can find the distance here. It is far away from the chelazal. It is nearer to the micropyle end. Okay. Now this one you will find very important, Megasporogenesis. This is also a 5 mark question with the diagram with explanation, and you can find many questions will be arise from this part. Okay, here with the diagram which they have shown, it is the embryo sac with the megaspore mother cell which we have shown earlier here in the previous one. What we have shown here, this in this ovule only this part has been enlarged, the center part only it has been enlarged, and it has been shown something like this. Okay, so this one is that megaspore mother cell, and surrounding to that you can find cells. These are what? Nucellus. What is their function? That is to provide nourishment for the growing embryo sac or for the growing egg cell. Okay, now this one cell which we call it as megaspore mother cell, if I am showing it here, we will draw something like this. You draw just neatly because it takes time. I am drawing just roughly. Okay, so this is the nucleus of it. So what we say, this one as nucellus, this one is megaspore mother cell. Megaspore mother cell, in short MMC. It can be microspore or megaspore, so you must make clarification either it is megaspore or microspore. Now this one towards this end they have given it as micropyle end, micropylar end. If you find in your textbook, it is very clear. Okay, so now after this what happens? This will proceed with the meiosis. Okay, it takes place with the meiosis 1. Now this megaspore mother cell, after undergoing meiosis 1, then what does it form? It results with the formation of 2 cells and covered by the nucellus, same as you can find it here. Okay, so 2 cells are formed, so it is called as megaspore dyad. Okay, micropylar end. Earlier it was having only 1 cell. Okay, megaspore mother cell. Now it has become megaspore dyad after first meiosis. After second meiosis, the 2 cells will end with the formation of 4 cells. Is it? So this one is megaspore dyad. Okay, Megaspore dyad. Then it undergoes meiosis 2. After completion of meiosis 2, it forms 4 cells. Okay, it is called as Megaspore tetrad. Megaspore tetrad. Again, same, the nucellus and the micropylar will remain same, as you can find here. These are Megaspore tetrad. Tetra means 4, as you know, and dyad means 2. So first one Megaspore mother cell will divide by meiosis to form Megaspore dyad, and it will continue its meiosis too and results in the formation of Megaspore tetrad. Now in this Megaspore tetrad, 3 will be non-functional, only 1 will be functional. Okay, what happens to these 3 non-functional? It will get degenerate. That does not have any function, later that will get degenerate, only which is present towards the micropylar end. Here we said it as micropyle, is it? So the one which is present towards the micropyle end, it will continue its further division. Okay, now this one will get degenerate. They are non-functional, so they will get degenerate. Only one of the Megaspore tetrad will continue its further division. Okay, now one of that Megaspore tetrad, it continues with the mitosis. Okay, when it continues with the mitosis, how it appears? It appears something like this. Okay, now this one cell which I have shown here, it has been elaborated and it is like this. Here I have shown only one nucleus, but here I have shown 2 nucleus. Why? Because it has undergone a division called as mitosis. Earlier it has taken place meiosis, right? This was a diploid. After meiosis, it will result in the haploid, right? Each one was that haploid. Now when it is undergoing mitosis, whether these are haploid or diploid, it is haploid. Okay, each one are haploid, because mitosis equal division. Here there is no reduction division. So when one nucleus will divide, here you can find I have not divided something like this. Why I have not divided? Because here first nuclear division takes place. Okay, later cytoplasmic division. So because first it takes place only nuclear division, so we are not showing this line. Just 2 nucleus will be formed, later cytoplasmic division will take place. For this we call it as 2 nucleated embryosac. Okay, it is called as 2 nucleated embryosac. Got it? Now you don't draw this line which I have shown. It should not be there, because it is only nuclear division, not a cytoplasmic division. Now again what happens? It will proceed with the one more mitosis. Okay, one more mitosis, the same cell. Now we have found from one nucleus we have got 2 nucleus after one mitosis. Now from 2 nucleus, how many nucleus you will get? You will get 4. You will get something like this. Again, no cytokinesis, only karyokinesis. Okay, only nuclear division, no cytoplasmic division. So you will find what will give the name for this: 4 nucleated embryosac. Okay, it is 4 nucleated embryosac. Now next what happens? Again in one more time it undergoes mitosis. When it undergoes mitosis, then what? These nucleus which are there, each one will divide. Okay, so from 4 you will get 8. Okay, you got something 8 nucleus, something like this. Okay, now this we call it as 8 nucleated embryosac. I am writing in short, so you can write it in full form: 2 nucleated embryosac, 4 nucleated embryosac, and then 8 nucleated embryosac. Now this 8 nucleated embryosac, later it becomes something like this. Okay, it will differentiate with the cytokinesis. Now cytokinesis takes place. Okay, now what happens? Roughly I am showing, it is there in your textbook with a neat diagram. Okay, so you can find this. How many, as we have given 8 nucleated embryosac here, it must consist of 8 nucleus. 1, 2, 3, 4, 5, 6, 7, 8. Total 8 nucleuses are there, and now it has been with the cell wall. So each one is having its cell wall. Okay, now these 2 are not having cell wall, that is having this complete itself will act as a one cell with the 2 nucleus. Okay, so now in this you can find this region I'll give it as chalaza, already it has been mentioned, and this one I'll call it as micropyle. Okay, but when I have started I have said upper one as micropyle and lower one will be chalaza, but now it has been inverted where micropyle is present and this side micropyle is present and this side it is chalaza. Now towards chalaza what three cells I have shown, those are called as antipodals. Okay, those are called as antipodals, and these are called as two polar nuclei. And the cell which is present here, it is central cell. Okay, here two polar nuclei are present where? Two polar nuclei are present in the central cell, and these three, in that two are synergids and one is X cell. Here I don't have a space, I shall show you here in the textbook. Okay, we'll come to that diagram. We shall start from the beginning. This was Megaspore mother cell which has undergone meiosis to form Megaspore dyad. Then it has undergone once again meiosis to form Megaspore tetrad. We said that three are non-functional, only one is functional. That one functional will proceed with further division to undergo mitosis to form Megaspore, that is to form 2 nucleated embryo sac. Again it undergoes mitosis to form 4 nucleated embryo sac. Once again mitosis to form 8 nucleated embryo sac. Okay, now this 8 nucleated embryo sac, it will start to rearrange itself and it will become something like this. Okay, this is that structure. The actual structure, the mature embryo sac will be something like this. Now here as you can find, this is a chalaza end and this is a micropyle end. Here you can see they have shown first here micropyle end, opposite to that will be chalaza, but here they have inverted this micropyle end which was upper side, now it has come to the lower side and the other end is chalaza end. Okay, now in the chalaza end what the three cells are present, those are called as antipodals, and the one which is present towards the micropyle end, three cells are present towards the micropyle end, and in those three cells two cells are synergids and one cell is X cell. Okay, so three are present towards the micropyle end, three cells are present towards the chalaza. The cells which are present towards the chalaza are called as antipodals, and the cells which are present towards the micropyle, they are egg apparatus. All three together we call it as egg apparatus. Egg apparatus consists of two synergids and one X cell. Okay, in this synergids you will find there is presence of filiform apparatus. You can find there are small finger like structures. These filiform apparatus, it helps in the guiding of pollen tube. It will help that pollen tube to penetrate from this direction. Pollen tube will enter from this direction. Okay, then you can find there is presence of central cell. This portion is the central cell, and in this central cell there is presence of two polar nuclei. Okay, two nuclei are present. For this what we call it as 7-celled 8 nucleated embryo sac. 8 nucleus we said. How come 7 cells? See, there are 3 nucleus, 3 cells. Here also 3 nucleus, 3 cells. Total 6. Now this is one cell carrying 2 nucleus. Okay, so 2 nucleus are not present in 2 different cells as you can find here. They are present, 2 cells are present in one, 2 nucleus are present in one cell. So 3 plus 3 plus 1, it is 7 cell, and 3 plus 3 plus 2, it is 8 nucleated. So we call it as 7-celled 8 nucleated embryo sac for this diagram. Most of the time they have asked to draw a neat label diagram of this for 2 marks, only to draw this mature embryo sac or 7-celled 8 nucleated embryo sac. You have to draw this one. Okay, this one will be asked only to draw or explain. Many of the times for explanation also it has been asked. Okay, you have to explain this. Explain Megasporogenesis or explain the formation of mature embryo sac, then you have to start from here and end till here. Okay, depends on the question what type of question they have asked. Explain Megasporogenesis starts from here and till here. Explain formation of mature embryo sac starts from here and still here. Explain the diagram of mature embryo sac, then this will be it. Only this diagram you have to draw and explain it. Okay, now because we said here only one megaspore will continue further division, the rest 3 will get degenerate, because from one megaspore embryo sac has been formed. It is called as monosporic development. You can find in your textbook, it is here. Okay, in a majority of flowering plants one of the megaspore is functional, only one of the megaspore is functional while the other 3 will degenerate. Only the functional megaspore develops into the female gametophyte. This method of embryo sac formation from a single megaspore is called as monosporic development. Okay, we have got what is this embryo sac formation monosporic, why it is called as monosporic development. Okay, why female gametophyte is called as monosporic development. The method of embryo sac formation from a single megaspore is called as monosporic development, because it would have been formed from all the four, but it has formed only from one single megaspore. That's why it is called as monosporic development. Okay, now for explanation of this, you can start from here, starting from my Megasporogenesis. In Megasporogenesis, where you can find the definition of it, and then you can continue. You can give all this explanation, and some questions are there, some points, refer that figure, that this they have given. No need to write all those. Then you can get the information something like this till here. Okay, now in this will be complete explanation for your 7-celled 8-nucleated embryo sac. Here they have given the egg apparatus. The egg apparatus consists of 2 synergids and 1 egg cell, and the synergids have special cellular techniques at the micropolar tip called as Filiform Apparatus. Their role is guiding the pollen tubes into the synergid. 3 cells at the chelazal end are called as antipodals, and the last central cell is called as 2 polar nuclei. So these synergids are also called as help cells. Another name of synergid is help cells, HELP, help cells. Okay, because they are useful in helping or guiding the pollen tube. Okay, so this will be the explanation for your structure of the embryo sac, or it can be explanation for complete megasporogenesis. Okay, so this is about today's class. If you have any doubts, any queries, please comment it in the comment box. We shall meet on next video. Stay safe, stay healthy.